All right, today we are going on a mission. Think of it as a molecular heist. Our target? Just one specific protein. And the vault? A super dense, complex piece of biological tissue. The real challenge is, how do we snag just one type of molecule from literally trillions of others? Let's get into it.
So yeah, that's really the big question, isn't it? How in the world do you find one specific molecule among trillions? It's kind of like trying to find one particular grain of sand on an entire beach. Inside a cell, our little target protein is just swimming in this sea of thousands of other proteins, not to mention fats, sugars, you name it. So how do we pull this off? How do we get in and out with just our prize?
And just to give you a sense of the scale we're working with here, take a look at this. You can see one protein molecule, that little blue dot, right next to a much larger enzyme. We are operating in a world that is just unbelievably tiny. Every single step of our plan has to be incredibly precise. Okay, so here's the plan, our heist, if you will. First, we've got the smash and grab, basically breaking into the cell. Then comes the really tricky part, sifting through all that molecular chaos. After that, we need a final purity test, to make sure we actually got the right thing. And then finally, we get to examine the prize itself. We're going to decode the protein's blueprint and figure out what makes it so valuable. All right, so every good heist starts with a break-in, right? Well, ours begins with a pretty brute force approach. The vault is beef heart tissue, and it's absolutely packed with mitochondria. Those are the tiny power plants of the cell. We need to crack that vault wide open to get to all the molecular treasure hidden inside.
This is our 5-stage purification process. And you can see, it really does start out messy and gets more and more refined as we go. Think of it like going from a sledgehammer to a delicate lockpick. We're going to slowly separate our target protein from all that other cellular noise. Let's kick things off with stage 1. Step 1 is homogenization, and honestly, it's basically what it sounds like. We're putting the beef heart tissue into a really powerful blender. Now, we do this in a special solution that's cold, buffered, and contains sucrose, and there's a reason for all that. The cold temperature, that keeps our target protein stable so it doesn't just fall apart. The buffer keeps the pH just right, and the sucrose, that's to keep the organelles, like the mitochondria where our protein is hiding, from bursting. Pretty clever, right?
So after blending, we're left with this thick, soupy mix of all the cellular components. To start sorting through this mess, we use a centrifuge. What this does is spin the mixture at incredibly high speeds. All the heavier stuff, like cell nuclei and any unbroken tissue, gets forced down into a solid pellet at the bottom. We just care about the liquid that's left on top. That's called the supernatant, and that's where the smaller mitochondria and our target protein are floating around.
Okay, so we've broken in, we've grabbed the loot, but it's still in a big bag mixed with a whole lot of other junk. Now it's time for the really sophisticated part of the heist. Let's move on to the more delicate, precise work. Now, this next technique is really cool. It's called salting out. What we do is add a very specific amount of a salt, ammonium sulfate. This makes some of the proteins we don't want become insoluble. They basically crash out of the solution. We spin those down and just toss them. Then, we add even more salt to the liquid that's left. And this new concentration is perfectly calibrated to make our target protein precipitate. So now we can collect it as a nice little pellet. So we've got our protein, but it's kind of covered in salt. The next step is really just a cleanup phase called dialysis. We take our redissolved protein pellet and put it into this special bag that has microscopic pores. Then we stick that whole bag in a big container of buffer solution. The tiny salt molecules, they can just pass right through the pores and float away. But our protein is way too big, so it stays trapped inside the bag, nice and clean.
All right, now for some real finesse. We're going to run our cleaned up sample through a column that's packed with these tiny, porous beads. The technique is called size exclusion chromatography, and you can really just think of it as a molecular obstacle course.
So in this obstacle course, the smaller proteins get caught up inside all those little porous beads, which means they take a much longer winding path to get through the column. But the larger proteins, they can't fit into the beads at all, so they just flow right around them and come out the other side first. And our protocol specifically tells us to collect the very first liquid that comes out. So what does that tell us? It tells us our target protein is one of the bigger molecules still left in the mix. That's a huge clue.
We're getting so close, but we're not quite done. The next step separates proteins based on a completely different property. Not their size, but their electrical charge. For this, we use a column that's packed with negatively charged beads. It's called cation exchange chromatography. Any positively charged proteins are going to stick to those beads like a magnet, while anything negative or neutral will just flow right on through. So at the pH we're using, our protein is positively charged, which means it's now stuck to that column. So, how do we get it off? Well, this is the clever part. We wash the column with a solution that has a higher pH. This completely changes our protein's charge. It neutralizes it. And once it's neutral, it just lets go of the negative beads and flows right out of the column. It's a brilliant way to specifically target and release our molecule, leaving those last few impurities stuck behind.
Okay, the heist seems to be complete. We've got the goods. But in science, just like in any good heist movie, you have to verify what you've got. The job isn't over until we can prove, without a doubt, that we have exactly what we came for and absolutely nothing else.
So, our first check is a technique called isoelectric focusing. It separates proteins based on their isoelectric point, or pI. That's just the exact pH where a protein has no net electrical charge. It's like a unique fingerprint. But when we run our sample, we see 3 distinct bands. What does that mean? It means we actually have 3 different types of proteins in our sample. We're not as pure as we thought we were.
But we are not giving up now. Our protocol tells us our target is the band at pi 5.6, so we literally cut that single band right out of the gel. Then we take that tiny slice and run it on a second, completely different type of gel called SDS-PAG, which separates proteins by their size. And this time, we get one single, clean band. That's it. That's our protein. Pure and finally isolated. The heist is a success.
So, we pulled it off, we have the protein. But what was the whole point of this incredibly elaborate heist? The true treasure isn't just the molecule itself, it's understanding its structure. The real prize is decoding the blueprint that gives this molecule all its power. That knowledge is fundamental to, well, everything, from understanding diseases to designing brand new medicines.
When scientists first managed to visualize a protein's 3D structure, they were just floored. As the Nobel laureate John Kendrew wrote back in 1958, the arrangement was so much more complicated than any theory had predicted. It had this lack of symmetry that just defied all their expectations. Proteins aren't simple, predictable little machines. They are these incredibly intricate, beautiful pieces of molecular architecture.
And this right here is the central secret. A protein's function, what it does, is determined entirely by its unique 3-dimensional shape, its conformation. And that unbelievably complex shape is dictated by nothing more than the linear sequence of its building blocks, the amino acids. If you change the sequence, you can change the fold, and ultimately, you change the function. It all comes back to that.
So how does a simple chain of amino acids fold itself into such a ridiculously complex shape? Well, it mostly comes down to 2 simple rules driven by basic physics. First, any parts of the chain that are hydrophobic, meaning they hate water, they bury themselves deep inside the core of the protein to get away from the water all around them. And second, the structure folds in a way that maximizes the number of stable hydrogen bonds that can form on the inside. All these tiny weak forces acting together create a strong, stable, and functional structure. And finally, the very backbone of the protein itself provides a huge structural constraint. The link that connects the amino acids together, it's called a peptide bond, is rigid and flat. It can't just rotate freely. This means the protein backbone is kind of like a series of stiff, flat plates connected by flexible hinges. This limitation dramatically cuts down the number of possible shapes the protein could be in, which helps guide it into folding into its one correct functional shape.
Okay, so we've established that a protein's specific folded shape is absolutely essential for its job, which leaves us with one last, really critical question. If everything a protein does depends on its intricate structure, what happens to its function when it loses that shape?